molecules if I say to you Aristotle's spontaneous generation? So we introduced the past last time, and now it comes back to mind. All right, so chapter 3, I just want to lay down some basic tools. Talk about some basic things we can do to look at bacteria, right? So keep in mind, we talked about gram stains in the lab last week. Today we're going to get back to that a little bit. So I want to define some general terms for you: prokaryotes and eukaryotes. And I want to talk about something called a binomial system of classification. So let's go through these four. And again, they're very simple concepts. If you take a look at all of life, we can actually classify them in various ways. The first classification system looks like this. We actually place them into 5 kingdoms, all right? So we say here's animals, here's plants, here's fungi, here's proteins. This is like a kind of strange one. These are single cellular organisms. And the bacteria back here, they used to be classified, or they are classified as monomers. So this is one way to look at classifying life. We have 5 different kingdoms. 1, 2, 3, 4, 5. So we go monomer, protista, animalia, animals, which include us, fungi, and then plantae. Of course, we're going to focus on bacteria only today, right? Now, there's another way to classify life. So we just saw the 5 kingdoms. And back in the 70s, a guy named Wusi said, you know what, if you look at nucleic acid sequences, we can classify all of 5 into 3 different domains. So we have the first one I just showed you, that's based on kingdoms, right? This other classification system is based on what we call RNA sequences. So we have the domain bacteria, domain archaea, domain eukarya. So both of these, if you recall, bacteria and archaea, they are prokaryotes. So remember, prokaryotes are defined by this. They don't have nuclei. They don't have organelles. So a bacterial cell does not have nucleus. Your cells do, all right? Archaea and bacteria, they are prokaryotes. You see that right here. Everything else in life is eukaryotes. If I look at plants, if I look at animals, if I look at protists, in other words, the other 3, they are eukaryotic cells. So eukaryotes have nuclei. All right. Now, if you look at the basic organization, just to give you a sense of what bacteria look like in comparison to a eukaryotic cell, and again, these are just drawings. So here's what your bacteria look like. So back here we have cell walls, and again, if you remember the lectures, the cell walls are made up of peptidoglycan. We have this coat right here in blue. That's called a glycocalyx, the cell wall, cell membrane, and the cytoplasm. But here's what I really want to point out to you. Right over here, that's the chromosome of the bacterial cell. So the chromosome right here, if you notice, there's not a nucleus. So that DNA is just in the cytoplasm, sitting, is draped in the cytoplasm. Now compare this to your cells. These are eukaryotic cells. So plant cells look like this, animal cells look like this, protists look like this. And the basic characteristic is right here I want to show you. There's a nucleus, right? So again, to summarize what I'm trying to tell you here, bacterial cells have a very different morphology compared to you cells. The basic difference is this. Bacterial cells do not have any organelles, meaning they do not have nuclei compared to your cell, right? So, again, we're not going to focus on eukaryotes. We're going to focus primarily on bacterial cells. So, just a few things about your cells, which are eukaryotes. Remember, they have nuclei, they have organelles. Organelles are what we call subcellular structures, things like mitochondria. You don't find these in bacteria. Endoplasmic reticulum, Golgi, lysosomes. These are all examples of organelles that you have in your cells, but you don't find in bacterial cells. Still think about this. Bacteria have to survive. They have to replicate themselves. So practically everything you do as far as processes go in eukaryotic cells, basically bacteria do them also, right? Now, chapter 4, which I started today, we're going to focus on a very few structures on bacterial cells as they relate to disease. All right. Okay. So bacteria can have flagella. Bacteria don't have cilia. Cilia is another type of filaments that cells use to move around, but they don't have this. Again, I'm not going to focus on eukaryotic systems, all right? I'm primarily on prokaryotes. Now, what I want to get to is this, really, microscopy. And first of all, before I get to microscopy, I just want to say just one little thing. the way we classify organisms and study them. So really, this is the important thing. All those slides, I don't want to say they're meaningless, but I'm not going to focus on them. I do want you to know this. Bacteria are prokaryotes. Your cells are eukaryotes. And again, we defined that like 2 or 3 times already. The basic difference is this. Bacteria do not have organelles. So if you leave this class and you say bacteria have nuclei, man, we didn't really learn it right? So again, that's all I want to get from those slides. Now, the next thing is to tell you this. When we study bacteria, we're going to give them 2 names. The system goes back to about 300 years ago. It's called a binomial system of classification. And again, this is not a class in history, but I do want to illustrate just a couple things for you, right? Again, if I were to say to you strep throat, I'm just thinking about a week or 2 from now, you should be thinking, hey, that's a strep disease. No, I want you to go a little further. You're going to say that disease is caused by an organism, a very specific organism called Streptococcus pyogenes. There are 2 names to it, all right? Just like the scientific name for humans is Homo sapiens. Well, for bacteria, we have 2 names also. And again, I don't want you to say staph or strep. I want you to give me the genus name and the species name. The first name, in this case, is Homo. The last name is sapiens. This is the genus name. And the second word here, sapiens, would be the species name. Now, let's put this together for bacteria. Let me give you an example. What you're looking at here is a disease called necrotizing fasciitis, right? The fancy name for it is necrotizing fasciitis, but you probably heard about a disease called the flesh-eating bacterial disease. Well, this is an example of it. Now, many different organisms or bacteria can cause this disease. But top of the list is this one here, Streptococcus pyogenes. Notice it's not strep, it's Streptococcus pyogenes. It's a very specific organism. So when we look at a binomial classification for bacteria in particular, we have 2 different names. And again, you're going to see when we start talking about diseases, I would always give you, most of the times, I'm going to say this is the name of the organism. So if I give you an exam, I don't want you to say staph infection or strep infection. I want to be very specific. What species is causing that disease? So the genus name, again, is the first word here. So this would be Streptococcus pyogenes is the species name. For Staphylococcus aureus, one of the diseases it causes is toxic shock syndrome. It can also cause this disease here, necrotizing fasciitis, right? But again, the point is this. The genus name is Staphylococcus, and the species name is aureus. Just one more example, tuberculosis. Tuberculosis, also called TB, is caused by Mycobacterium tuberculosis. So just to point this out again, the genus name is Mycobacterium, tuberculosis is the species name. All right. Now, when we take a look at infections, how do bacteria establish infections? Well, forget the slide for a bit. I want you to try to think about this functional. If I have a single bacterial cell, and imagine that bacterial cell is now in a wound, my wound just got contaminated. One cell can't cause disease, all right? But many cells, if they have a coordinated way of working together, can cause disease. So although we don't have to think about this in general, bacteria have to establish residency in wounds. They have to multiply. They have to get to the right numbers before they say, you know what? We have enough cells to produce enough toxins to cause disease. Now, the way bacteria communicate with each other is through a process called quorum sensing that was discovered about 20 years ago. Literally, if I have 2 Staph aureus cells, one saying to the next, hey, we got to grow. We have to multiply ourselves. How do they do that? They communicate very effectively through molecules, all right? Anyway, all I want you to know from this slide is this. Bacterial cells can communicate with each other, usually in a very species-specific way. So imagine I have Staph aureus and E. coli. Staph communicates with a cell. Staph aureus communicates with a cell. E. coli would communicate with someone's cell. They're competing to get that niche, all right? So initially what happens, the bacteria attaches. I might have very few cells, and again, few cells can't cause disease, but they're talking to each other chemically, biochemically, until they get to the right numbers. And when they get to the right number, they say, okay, we have an army now, we can actually cause a disease. That process of communication is called quorum sensing. Ultimately, when you start replicating the bacterial cells, the numbers, of course, increase. You form a biofilm. So biofilm in this case is just a collection of bacterial cells in one area. That's because they grew over time. They communicated with themselves through a process of quorum sensing to allow them, here's the thing, to get to the right numbers. Because if you don't have the right numbers, you're not producing enough enzymes or toxins or whatever you need to do to produce disease. All right? Okay. Now, here's another way to look at those kingdoms I mentioned to you earlier. I just have the slide here for you. If you're interested, you can look at it. We have bacteria, fungi, viruses. These are microorganisms, of course. The protozoans are unicellular. They're single-cell organisms. But I cannot want to spend too much time memorizing little facts like this. What I'm trying to get.
I have a question.
Yeah, go for it.
You were saying something about bacterial cells and how they can communicate, and then you mentioned two different cells. But can bacterial cells only communicate like to the same kind?
Yeah, because in general, yes. So try to think about it this way. Imagine I have two very different organisms. And let me go back to the example I used. Let's just say I have Staph aureus, right? And E. coli, and both of those cells got into my wound. So here's what's happening. Both of them can grow, don't get me wrong, but there's a competition that occurs. One species want to grow and take over the other one. Usually, both species grow to some level, but keep in mind, it's beneficial if I'm Staph aureus, it's beneficial for me if I just up my numbers, right? So the way they do this is produce these little chemicals that only their cell recognizes. So E. coli can only recognize its own molecule. Staph aureus can only recognize its own molecule. That process of communication is called quorum sensing. All right, are we good?
Yes, thank you.
Nice, all right. Good question, by the way. All right, now as we go to this next bit back here, here's my objective, all right? Some of these terms you already know because you've seen them before. For example, if I say to you aerobic exercise, you kind of relate to that. So these terms, they apply specifically to bacteria. And here's why they're so important. Because later on, when I talk about things like tetanus or anthrax or whatever, I say these organisms are aerobic or anaerobic or facultative. I want you to understand what that means right off the bat. All right? So if you notice, I don't have definitions here for them because I'm going to give it to you. And you're going to write it down somewhere. All right? That's really important because think about this. If I say to you tetanus, that organism is caused by Clostridium tetani. That organism is an anaerobic organism. In fact, oxygen would kill it. So it's important to understand those terms. So when we start talking about these diseases, and as I say to you, halophilic or cyclophilic, it already comes to mind. You know some basic characteristics of those organisms. So let's list them. And by the way, most of this, they're intuitive. For example, if I say to you thermophile, well, first of all, philic means to love. All right? So philic, a phile, the thermo refers to temperature. So basically, they love temperature, high temperature. So your body temperature is 37 degrees centigrade. Thermophilic organisms, typically they grow in the range from 40 to about 90 degrees centigrade. Some of them even grow, now check this out, some of them even grow in boiling water. So they're called hyperthermophiles. They don't cause disease in humans. Why? Because they require a very high temperature for them to even start growing. So generally, we don't have a problem with thermophilic organisms. Does not cause disease. A very, very few species can, but in general, no problem at all. Now, the other term is really important. Psychro, not cycle. Psychrophilic organisms, notice, they grow at low temperatures. So if you open up your refrigerator, you put your arm in there, it gets pretty cold, right? That temperature is between 4 to 8 degrees centigrade. There are some organisms that can grow in refrigeration. You guys probably all know this. Spoiled milk. Well, there might be some salmon, actually listeria in there. That's a psychrophilic organism. So the term psychrophile is an organism that can grow in low temperature. Now, another term is halophilic. Halo refers to salt. So these organisms love salt. So think about this. Where do you find a lot of salt? In seawater. So there are some organisms that cause some really nasty disease. Luckily, very few in the United States. For example, Vibrio vulnificus. That's a very nasty organism. We tend to get that by eating fish that's contaminated, saltwater fish that's contaminated with the bacteria. There's another one, Parahemolyticus, a very, very dangerous organism. Very few cases, by the way, but nonetheless, those organisms are they're halophilic. So a halophile, forget about the name of the species. I'm not interested in that yet. I just want you to know these definitions. A halophilic organism loves to grow in a salty environment. Now, the other one, of course, is self-explanatory.